Cool. Today we have a presentation by Dr. Kylle Roy. Kylle is an entomologist with the U.S. Forest Service, and her presentation is titled Rapid ʻŌhiʻa Death Entomology. Kylle, thank you for joining us today, and you may be able to start whenever you are ready.
Hello, everyone. Thanks to the HETF staff for having me. I'm going to start sharing my screen now. Okay. All right. My first image, I brought up this like cute little thing that I think Charlotte made from the ROD team because I really like the cute little beetle graphics. But I'm going to be talking today about rapid ʻŌhiʻa death entomology, which is kind of a big, broad topic. So I want to acknowledge all the different people that have contributed to this work. So rapid ʻŌhiʻa death entomology in general started in around 2015, 2016 by Dr. Curtis Ewing and Dr. Gordon Bennett. And I worked as a technician for them. And then the work followed me when I went to USGS under Dr. Carter Atkinson, Bob Peck, and also Kelly Janicki contributed a lot to the beginning work. And most of what I'll be talking about today is what I did for my PhD at Purdue under Dr. Matt Ginzel. And a lot of the work was contributed by Dan Dio-Micros and Ellen Dunkel. Oh man, my slides are stuck. Poor service computers. Okay, so I wanted to start off by talking about the HETF, introduce you guys. So we already heard the land acknowledgement. I think it's so cool, so I put it on my first slide. So to really introduce you to the two different HETF units, Pu'uwawa, the dry forest, and then Lapuhoihoi, the wet forest unit. And since this talk is for HETF, I did a little bit of research to see what we know about ROD and ambrosia beetles in the HETF systems. So good news, Pu'uwawa, there's, as far as updated as of yesterday, there's been no ROD detections in Pu'uwawa. There are ROD-associated ambrosia beetles in the area. So there's this article led by Conrad Gillette, and he was a postdoc in Dan Rubinoff's lab who left a couple of years ago, where he was working on, there's a suite of native Xyleborus ambrosia beetles that I'll actually get into at the end of my talk. But his project was, focusing on seeing or rediscovering really native ambrosia beetles. And so he did a small amount of trapping at Pu'uwawa, and he found that Xyleborus ferrugineus and Xyleborinus saxesenii were present at Pu'uwawa. And at Lapuhoihoi, so there's ROD there. So you can see all these red dots are trees that have tested positive for Ceratocystis lukuohia or huliohia. But we don't know much about the ambrosia beetle species there. I actually will be trapping in this unit in the next couple of months, so I'll have information. And just like reflecting on the work that's been done in the HETF, I think there's lots of room for figuring out what's going on with ambrosia beetles and ROD in both of the units. Okay, So first I wanted to introduce ʻŌhiʻa lehua. They're so important to the Hawaiian culture because they are, they're literally used to be everywhere. And so they were also everywhere mentioned in Mo'olelo and Mele. They are the kinolau of the god of war, Ku, and also Laka, the goddess of hula. Is so prominent, the use of the flowers and the liko in hula and lei making. The different parts of the wood are used for hale and canoe and ki'i, specifically, ki'i or images of ku as well. And they're also used in traditional Hawaiian medicine or la'au lapa'au. So ʻŌhiʻa lehua are also super important to the landscape. Within Hawaii, it covers 350,000 hectares. 250,000 of that is on Hawaiʻi Island. They're the first tree to colonize the lava flow. They're the, keystone tree species. So many other species depend on them to survive. They create Hawaii's watersheds. They can be found from sea level to tree line. They're just super important. So to introduce you all to rapid ʻŌhiʻa death, ROD is caused by two different species of Ceratocystis fungi, Ceratocystis lukuohia and Ceratocystis huliohia. So they were given Hawaiian names to translate to what, how they act. So Ceratocystis lukuohia was given the name ʻŌhiʻa destroyer. So it's much more virulent than Ceratocystis huliohia, and it's also more widespread. Ceratocystis huliohia was given huliohia, so ʻŌhiʻa turnover, and so that acts more like a canker. But ultimately, both lukuohia and huliohia will result in true death, although lukuohia is much more aggressive. So they both clog the transport of water in the functional part of the xylem, also known as the sapwood. And in order for ROD to spread, there needs to be some sort of wound. So to introduce you all to ambrosia beetles. So when I first started working on ROD, we didn't know if there were any insects involved with the spread of ROD, but based on other closely related diseases, we thought that, ambrosia beetles might be a good place to look. And so ambrosia beetles are given the name ambrosia because they farm their ambrosia. So just like the fruit salad, they farm their ambrosia fungi. And they tunnel into trees and plant their ambrosia fungi in the xylem, also where Ceratocystis lives. And when they're making their homes, which are called galleries, they kick out what we call frass into the environment. And the frass actually means little tiny macerated wood parts that are the same size as their little mouths. And also dead beetle parts and poop, basically, is what the comprises frass. And so the beetles that are specifically have been associated with ROD are in the tribe Xyleborini. And so the Xyleborini beetles, they have this haplodiploid biology where females are diploid and males are haploid. And so a single founding mother can actually start an entire generation on her own, which makes them really effective invasive species. And so these beetles in general will be attracted to stressed, dying, and sometimes even healthy trees. And so as a general rule, when trees are stressed and they can't breathe normally, there'll be this accumulation of ethanol product. And so this ethanol scent will go out into the environment and bring in, well, these ambrosia beetles have adapted to know that scent means that this is a good place for me to colonize a tree and plant my ambrosia fungi and farm it. So we found 4 different invasive ambrosia beetle species associated with ROD: Xyleborinus saxesenii, Xyleborus affinis, Xyleborus ferrugineus, and Xyleborus perforans. And so all 4 of these ambrosia beetles, I said they're invasive species, so they're not native to Hawaii. I listed all the origins of where they're from. They're all generalists, and so they have a really wide host range. And many of them have been associated with other ceratocystis diseases. So there's 2 ways that the ambrosia beetles can be involved with the spread of ROD. The first is called what we refer to as indirect transmission. So that is what we refer to as like the movement of the frass that they're creating. And so in this paper that was recently published, we were able to show that frass can contain viable Ceratocystis spores that can then go and kill ʻŌhiʻa seedlings. And we also looked at the spore type, and we found the spore type to be what are called chlamydospores, so they're just resting spores. And viable spores can survive for at least 6 months in the environment in frass, and that was just the limit of the study, so potentially they could survive much longer. So when you're coming into contact with ROD in the environment, you're most likely to come into contact with it in the form of frass, and that frass is what can get blown in the wind, spread in the soil, get on your boots or get on your tools. Yeah. And so the other way that ambrosia beetles are involved with the spread of ROD is through what we call direct transmission. And that's what we refer to when the beetles can actually directly vector ceratocystis on their bodies. So we were able to show in this other recently published paper that ambrosia beetles will attack healthy trees, apparently healthy ʻŌhiʻa trees, and we were able to show that they're more likely to attack trees that had been wounded. So that was simulating stress, and so they were coming into the stressed trees. And in areas where there's a lot of ROD, we hypothesized that that's where they'll act more like vectors, just because there's a lot of ROD in the area. And they're kind of will just like fly around aimlessly. And then Ceratocystis is such a virulent tree pathogen that, just a couple of spores can kill an entire tree. So a beetle just tunneling in to a healthy tree can kill the tree. And we found that one beetle species, Xyleborus affinis, was most likely to carry viable Ceratocystis spores on its body. So these are the 4 ROD associated beetles, and I kind of made this like summary slide for the things that we figured out over the years without having to show you guys a bunch of figures. And so again, Xyleborus affinis was most likely to carry the Ceratocystis propagules on its body. Xyleborus ferrugineus produces the most frass. They also colonize only the lower portion of trees, so the lower 3.5 meters of trees. And not to confuse anyone, but I figured I might as well start telling people we actually have their name incorrect. They are actually Xyleborus bispinatus. That is a work that Ellen Dunkel is leading. And so basically we have Xyleborus ferrugineus and Xyleborus bispinatus, both species. historically in Hawaii, but recent collections have all been Xyleborus bispinatus. So maybe just ignore that because I'm going to call them Xyleborus ferrugineus for the rest of this presentation. And also I wanted to add that Xyleborus affinis, Xyleborus ferrugineus, and Xyleborus perforans all appear to be limited by elevation. So they're found up to 750 meters. They can be found in higher elevations. It just depends on the rest of the host plants, the level of invasion in the area. And Xyleborinus saxesenii can be found, from, low elevation all the way up to high elevation. So taking a step backwards to the Ceratocistidaceae family. And so the Ceratocistidaceae family, where lukuohia and huliohia are, they are known to have co-evolved with insects for dispersal. So Ceratocistidaceae fungi, they produce these long-necked parathecia that push out these sticky ascospores. And those sticky ascospores can get attached to an insect's exoskeleton. These fungi also produce fruity and floral odors, and then insects will be attracted to that scent. So this coevolution between Ceratocistidaceae and insects has been heavily studied in the systems of oak wilt. So that is vectored by nididulid beetles, and then also Ceratocystis of plain or Ceratocystis platani, and the ambrosia beetles Platypus cylindrus. So the first little, I guess now really getting into the chemical ecology stuff. So the first little project chapter was identifying the volatile organic compounds associated with rapid ʻŌhiʻa death, and also looking into the behavioral response of these ROD-associated ambrosia beetles to the Ceratocystis volatiles. And so I did this in 3 parts. The first was collecting the fungal volatile organic compounds, or VOCs, and I used what's called solid phase microextraction. And so it's just this special needle that you can expose to a fungal culture. Try to get my mouse back. Mouse back. Okay. And so you just expose it to the fungal culture, and then the volatiles or the compounds get bound onto this tiny fiber, and you take that fiber and you analyze it by gas chromatography and mass spectrometry, or GCMS. And then you can identify the compounds. And so then we use the same SPEME technology for ʻŌhiʻa seedlings that were inoculated with Ceratocystis lukuohia or Ceratocystis huliohia. And then we also had just wounded controls that were inoculated with water. And so this is again using that SPEME technology. And so I exposed it to a little bag that was around the inoculation point. And then again, analysis with GCMS. And for the behavior, we used what is called a still air olfactometer. So it's basically just a little plastic box. It has 2 openings on the bottom. And so we would use one side with a blank petri dish, and then another side with a culture. This is a culture of Ceratocystis lukuohia. And then we did timed assays where this tiny little dot, if you could see it, that's ambrosia beetle. And then so we would time how long it spent over a blank or the fungal culture. Okay, so jumping right into the results, we found 9 compounds of volatile organic compounds in Ceratocystis lukuohia and 10 compounds in Ceratocystis huliohia. So both lukuohia and huliohia produced those general fruity odors, and they also both produced banana-like odors. The one compound that was different was that Ceratocystis huliohia produced a pineapple-like odor that was absent in Ceratocystis lukuohia. So we were able to find the same compounds both in culture and also in inoculated ʻŌhiʻa seedlings. Something that was a cool finding was that when we inoculated or the wounded control ʻŌhiʻa, so when they were wounded, we were able to detect that ethanol scent that we had been hypothesizing was bringing in the ambrosia beetles. We were also able to detect the fungal volatiles 1 day after inoculation, and so that was actually 9 days before we could see, before there were any sort of symptoms. Really, we didn't see any symptoms until about 21 days. And then so this is pretty quick compared to what you'd expect to see in the field too. And so that's an example of a potential capability, you know, once technology gets there, for early detection using volatiles. So going over the behavior assays, so we actually saw that Xyleborinus saxesenii was actually a little bit repelled from Ceratocystis lukuohia, and so we have to look into why that might be. We saw that both Xyleborus ferrugineus and Xyleborus affinis were attracted to Ceratocystis lukuohia in culture. This is interesting because lukuohia, ferrugineus, and affinis, they all have neotropical origins. And also Xyleborus ferrugineus has been shown to be attracted to cacao trees that were infested with Ceratocystis cacao funesta, which is closely related to Ceratocystis lukuohia. And for Ceratocystis huliohia, we saw that Xyleborus affinis and Xyleborus perforans were both attracted to Ceratocystis huliohia, and which is interesting, particularly for Xyleborus perforans, because both huliohia and perforans have tropical Asian origins. I'm also have this figure over here where you can see that was a ʻŌhiʻa tree that had been inoculated with Ceratocystis lukuohia. And so you can see the staining here, this dark coloration, and that's the movement of Ceratocystis within the tree. And we were marking all the ambrosia beetle attacks with this like blue paint here. And so you can see that we had field observations that were aligning with our lab observations or our behavioral data. Okay, so now to introduce you guys to attractants and repellents for managing ambrosia beetles. So in the field of entomology in general, you can usually we can use semiochemicals, or I guess you can just say scents, to manipulate insect populations. And a lot of times people will manipulate pheromones, but actually ambrosia beetles aren't known to use any pheromone communication, but in fact, they're actually using pheromones. So they're using scents from trees. So some examples of attractants that can be used for ambrosia beetles include ethanol and different combinations of ethanol and methanol. And basically, I've talked about that a lot now. So that ethanol will simulate that the tree is habitable, and that's a good place for them to plant their ambrosia fungi. And then there are, you can use semi-chemical repellents to repel beetles. So it's kind of the same thing as using like mosquito repellent, keeping them away. There's different compounds. We looked at 2 different compounds of verbenone and also methyl salicylate. And those compounds can signal that the tree is decomposing, is uninhabitable, not a good place for them to plant their ambrosia fungi. And we tested those compounds in what's called a SPLAT formulation. SPLAT stands for Specialized Pheromone and Lure Application Technology. And so that is made by a company called Iskatech. And so we tested one compound or one product called Splat Verb that just contained verbenone. And then we also tested a different product called Splat Beetle Guard. And so Beetle Guard has verbenone plus methyl salicylate. And the advantage of this splat formulation is that it's, this natural, environmentally friendly product. And so it's a beeswax based product that you can just put out into the field using a caulking gun. And it should last, the product should last for about 3 to 4 months, and it should fully biodegrade within a year. And I can tell you that it does fully biodegrade within a year because I've seen it. And so the first thing that we did was we conducted a trapping study. So we wanted to see if what was the best attractant for the ROD-associated ambrosia beetles. And we found that ethanol was the best attractant. And we conducted another experiment where we placed the attractants in conjunction with the repellents, and to see, if the repellents were reducing trap capture. And we're able to see that both Splat Verb and Splat Beetle Guard were effective repellents for all of the ROD-associated ambrosia beetles. So you can read about that work in this paper here. So unfortunately, Beetle Guard has a temporary experimental registration with the EPA. And so for the next step, that means in Hawaii, what that means specifically in Hawaii is that we weren't able to test it on, we were limited to a 1/4 acre to test it. So for the next experiment, when we actually wanted to test the repellents directly onto trees, we only tested the product Splat Verb. And so Splat Verb active ingredient is that verbenone compound. And so we tested Splat Verb on healthy ʻŌhiʻa trees, so to act as a preventative measure, prevent them from getting inoculated and ultimately getting ROD. And then we also tested the Splat Verb product on ʻŌhiʻa trees that had ROD, ultimately trying to reduce ambrosia beetles in the ROD-infested trees and reduce the amount of frass that they're producing and ultimately inoculum spread in the environment. So we also tested 2 different label rates. So we tested the low label rate at 72 grams, and we also tested the high label rate at 108 grams. And then we quantified the abundance of the active ingredient, verbenone, over time using, again, that gas chromatography mass spectrometry. And putting those together, we saw that the for trees that for healthy ʻŌhiʻa trees, so green leaf trees, using the low dose of Splat Verb is efficient and also the most economical treatment, and that costs about $13 a tree to treat. And then for trees that already have ROD, to prevent beetle attack, the high dose of Splat Verb may be better. So the high dose would cost about $20 a tree for the ROD trees, and ultimately reducing frass production. Now, to put both of those results together, a lot of times there will be a new ROD outbreak, and so I pictured or tried to make a picture here of a new tree that has been detected with ROD. It's brown here, and so a lot of times there's will be just a few individual trees that are surrounded by healthy ʻŌhiʻa trees. So the healthy ʻŌhiʻa trees are pictured in green. And so you could treat the ROD trees with a high dose, and then the healthy green ʻŌhiʻa trees with a low dose of splat verb. I also wanted to highlight some recent work by Dan Diomicros. And so he did this work for his master's from Hilo, the Tropical Conservation Biology Environmental Science program, which I am also an alumni of. And so he tested both splat verb and splat beetle guard as an alternative to tarping. And so the current ROD management strategy when a tree is detected to have ROD is to cut and fell the tree and then tarp it. Now, tarping, creates litter in the environment. It needs to be removed or replaced over time. It can also create a mosquito habitat, which we really don't want to be doing right now, especially in high elevations. His project was able to show that both splat verb and splat beetle guard were effective at deterring beetle attack to fell bolts. And there's kind of really this critical colonization period when beetles will go to the freshly cut wood, and they were able to protect the bolts for that critical colonization period. And he was also able to show that it significantly reduced the amount of frass that was put out into the environment post-felling. And now, how am I doing on time? Anybody? Nobody's even paying attention anymore.
It is 12:35.
Okay, perfect. All right, because this is my last little, well, it's not the conclusion, but this is the last little study, my fun study. So now I'm going to introduce you guys to the ambrosia fungi in Hawaii. It was like the last part of my dissertation, my fun project. And so I talked a little bit about ambrosia fungi, a little bit about native ambrosia beetles. So ambrosia beetles they plant and farm this ambrosia fungi, and they actually have special pockets called mycotangia or mycangia where they keep their fungal spores that they plant. And the Xyleborus beetles tend to have it pictured here. What they call dual preoral pockets. And there's actually at least 21 species of native Xyleborus beetles in Hawaii. I say at least because there's definitely more that haven't been described. Yeah. And according to this work by Cognado et al. In 2018, they arrived in the Hawaiian archipelago approximately 9.8 million years ago. So in order to isolate the ambrosia fungi, basically you would trap beetles, either trap them using traps or directly take them out of trees using a chisel and hammer, like what Ellen's doing here. Collect the beetles, perform a series of washes to clean any other fungi that might be on the outside of their bodies. We actually decapitate them so we can only and then squish their heads so that you can just culture the fungi from the mitochondria, and then plate them, make a fungal lawn, and then you keep plating and plating until you get a xenic culture, so a pure culture. And then we extract the DNA from the fungi, and in this case, we used several different markers: ITS, LSU, SSU, and beta tubulin. And so I looked at the ambrosia fungi of these these 5 native Xyleborus beetles, and I have them grouped here by their host plants. And so Hawaiiensis, ʻŌhiʻa, Pele, and Dubiosis will all host Olapa. Dubiosis is more promiscuous and will go to other trees as well. And then, of course, I looked at the ambrosia fungi of all the invasive rod-associated beetles. And then I didn't talk about Xyleborus similimus too much. Xyleborus similimus is a native ambrosia beetle that is endemic to Hawaiʻi Island. It appears that, so they host only ʻŌhiʻa, and it appears that they are disappearing as well. They used to historically have been found at all elevations where ʻŌhiʻa is, and now they're getting rare and only found at high elevations. And so these are the results of the invasive rod-associated beetles. So we found a bunch of new fungal species that need to be described, and we're working on describing them and giving them names. So one interesting thing was that we didn't find any Ceratocystis in their mitochondria. So even though they can carry Ceratocystis on their bodies, they're not actually carrying them in their mitochondria. But so culturing is very biased as to what grows and how quickly things grow and whatnot, what media you use. And so we're actually investigating this a little bit further by conducting metabarcoding on the rod-associated ambrosia beetles, and looking at so maybe there will be Ceratocystis in their mitochondria, and so it's this project that will be led by a UC Berkeley undergrad, Alex Boren, and we'll be working on it in a couple of weeks. Pretty excited about that. And so these are the ambrosia fungi that we found associated with the native Xyleborus beetles. Xyleborus dubiosis, I said, was a little bit promiscuous, so it goes to a lot of different host trees, and we think that is probably why we found so many different ambrosia fungi associated with it, a lot of new species. And let's see, Xyleborus hawaiiensis, ʻŌhiʻa, and Pele, they were actually all isolated from a single down Olapa tree. And so it's really cool that they all have totally different ambrosia fungi living, in galleries that are right next to each other, farming different fungi. And I just wanted to show you all one of the phylogenetic trees that we put together for Raphaelia fungi. And basically we found 4 putative new species of Raphaelia that are shown in blue. And I wanted to point out everything that we really know about Raphaelia fungi has been all based on stuff that has been isolated from related ambrosia beetle, Xyleborus glabratus. And so Xyleborus glabratus is the vector of laurel wilt disease, which has killed at least 320 million laurel trees in America. And so laurel wilt is caused by Harringtonia. Let me show you guys here, Harringtonia lauricola. And so It used to be Rafflesia loricola, but it has been renamed Harringtonia loricola, and so it's a new group, Harringtonia. And actually, in one of the ambrosia ambrosia beetles, I have found a closely related fungal species, Harringtonia aguacate. And so that was kind of, it was alarming to me that just from our, really limited sample size, we were able to find something so closely related to laurel wilt. And so in conclusion, we're able to find that both lukuohia and huliohia produce fruity fungal volatiles that can attract their beetle vectors. Our VLC data are groundwork for potentially early detection because we were able to detect volatiles before there were any symptoms in ʻŌhiʻa seedlings. We were also able to detect that ethanol scent in wounded ʻŌhiʻa. And so that 100% ethanol is the best attractant for the rod-associated beetles. Splat verb and splat beetle guard are both effective repellents for the rod-associated beetles, and they also have potential to be used as tarping alternatives. So we'll use the high dose of splat verb for rod trees, and then the low dose for healthy ʻŌhiʻa. We also saw that there was no reapplication necessary over the 16-week trial period that we conducted. I'm working on getting splat verb registered in Hawaii to be able to be used. And based on the little bit of ambrosia fungi research I did, we just really know nothing about fungi in Hawaii. And there's really is a need for early detection and monitoring of insects and pathogenic fungi. So we know how ROD is spread. We have tools, aka repellents, to manage ROD. And we know ʻŌhiʻa are resilient, and we know that there are resistant varieties. Dr. Mark Hughes is leading a project here at the Forest Service where they are finding ʻŌhiʻa that are resistant to Ceratocystis lukuohia. And actually he's giving a presentation about it next week, I think part of the High Sam series. But I am fearful that ROD is getting forgotten because ʻŌhiʻa, they're still dying. So Dr. Flint Hughes, also at the Forest Service, he has long-term monitoring plots that he's been tracking the death of ROD for death of ʻŌhiʻa for 10 years on Hawaiʻi Island. And in his plots that where Ceratocystis lukuohia is present, he is seeing an annual mortality rate of 10%, which equates that in 20 years, all the ʻŌhiʻa in those plots will be dead. And so I know that's like really depressing. But at the Forest Service, we're working on this big project together. It's going to be the first time that a whole bunch of the ROD scientists are collaborating. So we're going to be doing a landscape scale study. We're going to be trapping for the beetles, looking, Kylle Roy is going to be looking at ungulates, specifically probably pigs, looking at the dynamics of fencing, and Mark Hughes will be looking at, will be actually planting resistant ʻŌhiʻa into these plots. And I just got my invertebrate collection permits for the HETF Lapuhoihoi for this project. And with that, there's so many people that have contributed to all of this work over time. They are probably not even all listed here, but the people that have helped the most are pictured here. And with that, I will take any questions. No question.
Thanks,
So,
Kyle. There is a question in the chat.
Oh, okay, I see. Rado. Hi, I always see you on Facebook. But we've never met in person. So your question was, can Ceratocystis lukuohia and Ceratocystis huliohia infect other Myrtaceae or even other families of trees? Okay, wait, I'm gonna answer that first. So Lisa Keith's lab has started that work, seeing if they could infect other other trees. Oh, I am so sorry. I'm gonna send you the paper because there's a new paper that they have found lukuohia can infect something that's not in Hawaii. There's a new paper, I'll send it out to you, but I can't remember at the top of my head right now. But it's more like something that is possible. It's not actually happening naturally. And I'll totally send it to you. I know your Facebook. So if so, are there no beetle vectors to spread them? I don't know what that means. So and then I'll also just like add on to that, saying that that Mark is also working on testing like other, because there's multiple ʻŌhiʻa species, and so he's also working on expanding testing the other Metrosideros species and seeing if they can be infected with Ceratocystis lukuohia. Why don't the native ambrosia beetles spread ROD if it's just carried on beetle bodies and beetle frass? Okay, so there's one native ambrosia beetle, Xyleborus similis, and they can spread ROD through the frass that they create. I don't talk about them too much, and so I talked about them in this presentation, showing that they're disappearing. They're still found at, or only found at high elevations, only on Hawaiʻi Island. So I didn't really want to villainize them, those poor things. But yeah, they can create frass. We don't know if they can. We don't know if they could vector. We didn't focus on that for the vectoring study that we did. But the project that I'll be doing with the Berkeley student, we will be doing the metabarcoding to see if the Ceratocystis fungi can be present in their mesotangia or on their bodies. So we'll have an answer to that soon. How long have the ROD-spreading beetles been documented here in Hawaii? A long time. So as long as each different beetle species, has been documented from a different time. But the answer is kind of basically like ever since there's been somebody looking. So the original insect collectors have found basically all the ROD associated beetles, so they've been here for a long time. And I can give you the actual numbers too when I also send you that paper.
I just wanted to say thanks, Kyle. I always learn something new when I listen to your presentations, and can't wait to hear some of the names that you guys come up with for the new species that you found. I also, side note, it's also really cute to think of these little beetles as farmers. I was just thinking, whenever you said that they're farming them, I was like, oh, cute.
Yeah, little
But also not
farmers.
cute, but like cute.
The native ones are cute. They can
Yeah.
be cute.
So thanks for
Yeah.
coming on and doing the webinar, Kyle.
I just want to cackle that. It was a very informative presentation. I think I learned so much more than I even thought I was going to learn today.
Good. Yeah, thanks for having me. I will look up those papers and send it to you, Geraldo. My brain is mush right now. Actually, no, it's better now. Better than it was yesterday.
Well, I believe we have about 7 more minutes. Just checking the time, being aware of people's time. But it doesn't look like there's any other questions in the comment section. But if you all want to follow up with Kyle, you can reach out to us and we can connect you. Our next webinar is going to be in June with Kyle Austin presenting rediscoveries and presumed extinctions of Hawaiian moths. Again, mahalo, Kyle, for sharing with us today. And thank you everyone who came.
Thanks, everyone. Kyle's
Thanks,
awesome
Kyle.
too. We're working on something.
Yay,
I'll
I'm
present it
excited.
next year.
Sweet.